Okay, so we've been talking about DNA, and DNA is what makes you you. It's also what makes a rat, and it's what makes a turtle, and it's what makes a snake a snake. But we haven't talked about how the DNA actually makes every living thing look different. So that's what we're going to do in today's video. So we're going to see what actually goes on in your cells to make a rat different from a turtle, different from a snake, different from you.
Hello, this is Mrs. Kohler here for part 2 of translating DNA into proteins. So we're going to talk about how transcription works, how RNA is edited, and about how it's translated into proteins. So the first step in this is called transcription. Remember, a scribe copies things, so you're making an RNA copy of the DNA. So you're making what's called mRNA, also called messenger RNA. Now take note that the first letter in mRNA is lowercase, and that's correct. It's not supposed to be capitalized. It should be a lowercase m, but capital RNA. And that actually carries the genetic information from the nucleus out into the cytoplasm. You don't want the DNA to leave the nucleus. You want it to stay there so that it stays intact. So the DNA can make a whole bunch of mRNA copies and send them all out to the cytoplasm so that it can be making the same protein multiple times all at once. Now, the way that things get added onto mRNA is with an enzyme called RNA polymerase. Now, there was DNA polymerase that added nucleotides onto a strand. Well, RNA polymerase does the same thing for RNA. So it lines up the DNA strand and then puts the complementary base on it. But remember, when you're making RNA, wherever you would have had a thymine, you're going to get a uracil instead. So transcription. We're going to practice this real quick. I'm going to give you an mRNA sequence, and you need to figure out what the correct complementary sequence would be. So here's your DNA. AGTTGCAGT. So the complementary strand, A would normally pair with T, but remember, for RNA there are no T's, so you'll put a U instead. G pairs with C, T pairs with A like normal. You can have A's in RNA. T pairs with A, G with C, C with G. Here you have another A that would normally pair with a T, but because there's no T's in mRNA, you're going to put a U instead, and then G with C and T with A. Now, once that DNA is translated into RNA, it has to be edited. It's just kind of like editing. So there are these segments of DNA that are called introns. And introns are actually long stretches of DNA that are nonsense DNA. They don't actually code for anything. And you have to cut those out of the RNA before you can turn it into a gene. So during a process that's called RNA splicing, you take the introns out. So here's an example stretch of RNA. This diagram calls it pre-mRNA. But your RNA is up at the top, and the two dark red portions on the end are the exons. They're going to stay, but the middle section is an intron. So what your body does is it has special enzymes called spliceosomes. You don't have to know that, but it's a cool word. That actually cut out that intron and then stick the exons back in together. So that process of cutting out the intron and putting the exons back together is called RNA splicing. So after the introns are removed, the parts that are left behind are called exons. And the way to remember exon is the EX in exon is like the EX in the word expressed. So an exon turns into a gene and gets expressed, and they get spliced back together again. Now, after transcription and splicing, you now have to translate that RNA into a language of proteins. So that's done by a molecule that's called tRNA. And once again, that little T in the front needs to stay lowercase. And this is called transfer RNA, and it actually carries amino acids to the RNA and links them together to make a protein. So if you look at this picture here, you can see that up at the top is the mRNA, and there's the 3 sequence segment, that's a codon. And this tRNA has a little segment on it that matches up with that with the complementary bases. And it's got the amino acid at the tail end. So that little segment that links on to the mRNA is called an anticodon. And what it does is it finds the codon that it matches up with, and it sits there for a second and leaves its amino acid behind so that they can link together. And there's just another version of showing what's happening there at the bottom. You have the U pairing with the A pairing with the U, the C pairing with the G. So during translation, now this happens in ribosomes. Remember when we did the cell unit, ribosomes make proteins. Well, this is how the ribosomes make proteins. The ribosome actually has a spot to let the tRNA and the mRNA meet up so that the amino acids can be added to a chain. So your ribosome is the big pink blob, and you have here tRNAs delivering their amino acids. Now the ribosome does have its own RNA called rRNA, but we're really not going to study that right now. If you take AP Bio, you'll probably talk about it, but that's not involved in translation. So, step by step. Number 1, the ribosomes are going to find the start codon, which remember is AUG, and that's where translation is going to start. It's going to bring in a tRNA that contains methionine to add the first amino acid, and then it's going to go down 3 blocks at a time to bring the other amino acids in order, and then when it gets to one of the 3 stop codons, it's going to stop making the protein, and it's not going to do any more translation. So then right here you have a diagram of the whole thing. This long black ribbon is your mRNA. You have 3 tRNAs delivering amino acids to the chain, and then this chain coming off up at the top here is your chain of amino acids that's eventually going to turn into a protein. Have a great day. Don't forget to write your podcast question. See you in class. That was my husband burping, just so you know. So shh. Just be quiet. So